Hi guys, so welcome to um, the high school biology lesson. So last time we talked about Darwin's theory of natural selection and how his theory rests on the idea that there exists tremendous diversity in any gene pool. We talked about how his theory picks traits that are more advantaged towards survival. Although Darwin could not explain the origin of diversity he saw in different populations, we now understand the process. So the source of variation in a population are there are three different sources of variation. So the first one is mutation. So mutations are changes in genetic material and are the raw material for evolutionary change. Raw material for evolutionary change. And next we have genetic drift. Genetic drift is change in the gene pool due to chance. So there are two examples of genetic drift. We have bottleneck and we have the founder's effect. So let's look at those two examples. So environmental disasters, that's right here, environmental disasters. So this is for bottleneck effect. Environmental disasters. So basically, environmental disasters such as fire, earthquake, and flood reduce the size of a population non-selectively, and it results in a loss of genetic variation. So the resulting population is much smaller and not representative of the original one. So certain alleles may be under or overrepresented compared with the original population. So this is loss of um, results in loss of um, genetic variation. So that's bottleneck effect. Founder's effect is established by a very small number of individuals from a large population. So established by a small number of um, individuals from a large population or larger population. So rare alleles may be overrepresented in this case. An example of a founder's effect is an incident that occurred in the old order of Amish of Lancaster, Pennsylvania. So they all descended from a small group of settlers who came to the United States from Germany in 1770s. Apparently, one or more settlers carried the rare but dominant gene for polydactyly, which is a gene that codes for having an extra finger and toes. So it's polydactyly. And due to the isolation and their small community of intermarriage, this population has a high incidence of polydactyly. So if you see this picture right here, we have an extra set of toes or an extra set of fingers. Now, going back to the sources of variation, we have last we have gene flow, and this is the movement of alleles into or out of a population. Into or out of a population. So it can occur as a result of a migration of fertile individuals or gametes between populations. For example, pollen from one valley can be carried by the wind across a mountain to another valley. Now, Hardy and Weinberg are two scientists who developed a theorem that described a stable, non-evolving population. In other words, they developed a theorem in which allele frequency does not change. So, frequency does not change. So, for example, if the frequency of an allele for a particular trait is 0.5 and the population is not evolving, then in 1,000 years, the frequency of the allele is still 0.5. This is called the Hardy-Weinberg equilibrium, and according to Hardy-Weinberg, if the population is stable, it must include 5 rules, which we will go over right now. So, in order to meet the Hardy-Weinberg equilibrium, first the population must be very large. So, population must be very large. In a small population, the smallest change in the gene pool will have a major effect on the allele frequencies. So, in a large population, though, a small change in gene pool will not cause a major effect and will be diluted by other individuals. Second, the population must be isolated from other populations. So there must be no migration of organisms into or out of the gene pool because that could alter allele frequencies. Third, we have there must be no mutations in the population. No mutation in the population. So a mutation in the gene pool could cause a change in allele frequency or introduce a new allele. Fourth, mating must be random. So if individuals select mates, then those individuals who are a better fit will have a reproductive advantage and the population will evolve. And we should not have evolving population. And lastly, there must be no natural selection. So natural selection causes changes in relative frequencies of alleles in a gene pool. Although having the trait that will give us the most benefit is maybe we would like that more in this case, but it does not apply to the Hardy-Weinberg equilibrium. So the Hardy-Weinberg equilibrium allows us to calculate frequencies of alleles in a population. So for example, scientists use letters, the letter P to stand for, they use the letter P to stand for the dominant allele. and then they use the letter Q to stand for the recessive allele. So the Hardy-Weinberg equation can be expressed as P plus Q equals 1, or P squared plus 2PQ plus Q squared equals 1. So this equation right here is derived from the monohybrid cross. And I'll show you guys why in a second. So let's draw a monohybrid cross, and we can say that has alleles of this and that. So they're both heterozygous, and let's cross them. We have this, that, and this. So if you look at this cross, we can see that there is two of the homozygous dominant, and there is one of there's actually two of heterozygous alleles, two of A, and then there is one of the homozygous recessive. So Q equals A. So we could see that there is one of homozygous dominant, two of heterozygous, and one of homozygous recessive. So this is how the equation of this is derived. Okay, so next we have forms of isolation. So a species is a population whose members have the potential to interbreed in nature and produce a fertile offspring. Anything that fragments a population and isolates small groups of individuals may foster the formation of new species. This is true because isolated populations are subject to different selective pressures in their respective environments. If enough time elapses, the two populations may become so different that even if they were brought back together, interbreeding would not occur. At this point, you can call that there's a completely different new species. So there are 6 different forms of isolation that commonly causes a new species to form. First, we have geographic isolation. Geographic isolation occurs when species are separated. So let's say mountain ranges, canyons, rivers, lakes, or glaciers may cause significant isolation between species. Next, we have polypoid. So this is a type of mutation that results from error during meiosis. Type of mutation that occurs during that results from errors of from meiosis. And um, yeah, and instead of being haploid or diploid, polyploid organisms can be tetraploid or octoploid. So polyploid organisms cannot breed with organisms that are not polyploid and therefore are isolated from them. And next we have habitat isolation. Habitat isolation. Habitat isolation. So this occurs when two organisms live in the same area but encounter each other rarely. So live on the same area but encounter rarely. So two species of one of the snake can be found in the same geographic area, while one inhabit the water, while the other one is mainly on the land. So basically, they really don't have a chance of meeting each other because they live in two different habitats, even though they live in the same area. Next, we have behavioral isolation. So, behavioral isolation occurs when two animals become isolated from each other because of some change in behavior by one member or a group. Here's when become because of change in behavior. So, for example, male fireflies of various species signal to females of their kind by blinking lights or their tails in a particular pattern. Females respond only to characteristics of their own species, flashing back to attract males. Next, we have temporal isolation. So, temporal refers to time. And yeah, this basically time. For example, different plants of one species living in the same area may become separated into two populations because some plants become sexually mature earlier and begin flowering the cooler part of the season, while other plants flower in the later, warmer part of the growing season. So they become maybe seasonally isolated and timely isolated. I'm going to use another slide to discuss the last one. And six, we have, finally, this is the last one, we have um, reproductive isolation. Reproductive isolation. So this refers to when closely related species may be unable to mate because of anatomical incompatibility. So, closely related species may be unable because of anatomical incompatibility. So, for example, a small male dog and a large male dog cannot mate because of their enormous size differences. So that is where I'm going to end it. So let's look over what we have learned today. So first, we talked about sources of variation. We have three, which are mutations, genetic drift, and gene flow. Mutations are changes in genetic material and raw material for evolutionary change. And then we talked about genetic drift, which is a change in gene pool due to chance. And we looked at two examples, bottleneck and founder's effect. And we talked about how bottleneck effect results from an environmental disaster such as fire, floods, and earthquakes. And this results in a loss of genetic variation. And then we talked about how founder's effect is established by a small number of individuals from a larger population. And we discussed how a small Amish community has a higher chance of getting this gene mutation polydactyly and having more, let's say, 6 fingers. And then lastly, we talked about, and the source of variation is the gene flow. And this is when the movement of alleles into or out of a population. Then we talked about the two scientists who found out an equilibrium and an equation. So Hardy-Weinberg equilibrium states that allele frequency does not change. And in order for this to be met, there has the population must meet 5 important criteria. So first, we have population must be very large. Then we have populations must be isolated from other populations. Then no mutations in the population should be present. And then mating must be random, and there should be no natural selection. Then we talked about the Hardy-Weinberg equation. Talked about how P expresses the dominant allele and Q expresses the recessive allele. And there's two equations, P plus Q equals 1, or P squared plus 2PQ plus Q squared equals 1. And we justified this equation by using the monohybrid cross. Then last thing we talked about is the forms of isolation. So first, geographical isolation is when species are separated. And then polypoid is a type of mutation that results from errors from meiosis. Then we have habitat isolation, which means they live on the same area but encounter each other very rarely. And then we have behavioral isolation when two animals become isolated from each other because of the change in behavior. And then temporal isolation is when the two basically don't meet because of time difference, time when they grow and time when they don't. And then finally, we have the reproductive isolation, which is closely related species may be unable to mate because of anatomical incompatibility. So that is it for today's lesson. And if you guys have any question, please comment in the comment section, and I'll see you guys next week.